CRISPR-Cas9, which stands for Clustered Regularly Interspaced Short Palindromic Repeats and CRISPR-associated Protein 9, is a groundbreaking gene editing technology that has, honestly, just revolutionized molecular biology. It holds immense promise for a whole range of applications, especially in medicine and agriculture. So, how does it work? Well, at its core, CRISPR-Cas9 is actually adapted from a natural defense mechanism found in bacteria and archaea. These microorganisms use CRISPR systems to protect themselves from invading viruses called bacteriophages by remembering their genetic code and destroying them if they show up again. Here's a simplified breakdown of how CRISPR-Cas9 works as a gene editing tool. Guide RNA, or gRNA, is super important here. The system relies on a synthetic guide RNA which has two key parts. First, there's a short custom-designed sequence about 20 bases long that's complementary to the specific DNA sequence you want to target and edit in a cell's genome. Second, there's a scaffold part that binds to the Cas9 enzyme. Cas9 is a CRISPR-associated protein that acts as a highly precise molecular scissor. It binds to the gRNA and, this interaction is what gives it the ability to target specific DNA sequences with such remarkable accuracy. So, when the gRNA Cas9 complex is introduced into a cell, the gRNA basically guides the Cas9 enzyme to the specific target DNA sequence in the genome by base pairing with it. Now here's a key detail. The Cas9 enzyme also needs a short DNA sequence called a protospacer adjacent motif, or PAM, to be present right next to the target site. This PAM sequence, it helps Cas9 confirm it's at the correct location. Once the gRNA finds its complementary sequence and the PAM is recognized, Cas9 makes a precise double-stranded cut in the DNA at that targeted location. After Cas9 cuts the DNA, the cell's natural DNA repair mechanisms kick in. Scientists can actually exploit these repair pathways to introduce desired changes, which is pretty fascinating. Non-homologous end joining, or NHEJ, is what we call an error-prone repair pathway, where the cell tries to rejoin the cut DNA ends directly. This often results in small insertions or deletions, also known as indels, at the cut site. And this can lead to knocking out or inactivating a gene. Homology-directed repair, or HDR, works a bit differently. If a repair template DNA sequence, one that contains the desired modification, is provided along with the CRISPR-Cas9 system, the cell can use this template to accurately repair the cut. This approach allows for precise knock-ins of new genetic material, correction of specific mutations, or even the insertion of entirely new genes. HDR is less efficient than NHEJ, and honestly, it typically occurs only during certain stages.
So why is CRISPR-Cas9 considered revolutionary? Well, let's dig in and find out what sets it apart from other gene editing technologies. CRISPR-Cas9 has garnered immense excitement due to several key reasons. First, its precision. This tool can actually target and edit specific DNA sequences with really remarkable accuracy. Then there's its efficiency. It's more effective than previous gene editing methods, which is honestly a huge leap forward. The simplicity and ease of use are also major factors. The system is relatively straightforward to design and implement, especially when you compare it to older technologies like ZFNs and TALENs. Plus, it's cost-effective, which means it's significantly cheaper and, much more accessible to researchers across the globe. And finally, its versatility stands out. CRISPR-Cas9 can be used to modify genes in a wide variety of living cells and organisms, including bacteria, plants, animals, and even human cells. The applications of CRISPR-Cas9. The applications of CRISPR-Cas9 are vast and continue to expand. Medical research, disease modeling. Creating cell and animal models with specific genetic mutations to study diseases like cancer, Alzheimer's, cystic fibrosis, and sickle cell anemia, helping to understand disease mechanisms and test potential therapies. So, gene therapy is all about correcting genetic mutations that cause inherited diseases. Right now, there are actually clinical trials going on for conditions like sickle cell disease, beta thalassemia, and even some forms of blindness. Drug discovery involves, identifying new drug targets and screening for drug sensitivity. It's a crucial step in developing effective treatments. Cancer immunotherapy is, about engineering immune cells, like T cells, so they can better target and destroy cancer cells. This approach is really changing the way we think about treating cancer. When it comes to infectious diseases, scientists are actually developing strategies to eliminate harmful pathogens by targeting their genes. For example, they're working on ways to remove HIV from infected cells, or even making mosquitoes resistant to malaria. It's a pretty exciting area of research with huge potential. Agriculture, well, that's another field where gene editing is making a real impact. Crop improvement is all about developing plants with enhanced traits. Things like disease resistance, higher yields, better nutritional content, and the ability to tolerate tough environmental stresses, such as drought or salinity. These advances could honestly help ensure food security for the future. Animal breeding is all about creating livestock with, desirable traits. Basic research? It's really the foundation for understanding genetics and how everything fits together. Studying gene function often means knocking out or knocking in genes, just to figure out their roles in various biological processes. Functional genomics uses high-throughput screening to, identify genes involved in specific cellular pathways or disease progression. While incredibly powerful, CRISPR-Cas9 also raises significant ethical considerations. Off-target effects. Despite its precision, CRISPR-Cas9 can sometimes make unintended cuts at other locations in the genome, leading to potentially harmful mutations. Ongoing research aims to improve specificity and reduce off-target edits. Germline editing. This is the most contentious area. Editing genes in human egg or sperm cells or in embryos would result in heritable changes passed down to future generations. This raises profound ethical questions about safety and unintended consequences. The long-term effects on future generations are unknown and, irreversible. Informed consent is, a fundamental principle in medical ethics, but future generations simply cannot consent to changes made to their genomes. The possibility of using CRISPR for, non-therapeutic enhancements could actually exacerbate social inequalities. It might even lead to a new form of eugenics, where only the wealthy can afford genetic improvements. There are concerns that eliminating certain genetic conditions could actually reduce human diversity, and this might, in turn, impact disability communities in ways we haven't fully considered. As a revolutionary but honestly potentially costly technology, CRISPR-Cas9 could end up worsening existing inequalities if access to its benefits is limited to only certain populations or countries. In agriculture, there are definitely concerns about the potential for genetically modified organisms to impact biodiversity, or even to create unforeseen ecological imbalances that could have lasting effects. Some ethical and religious viewpoints, express real concern about directly manipulating the fundamental genetic blueprint of life, sometimes described as playing God. CRISPR-Cas9 is a powerful gene editing tool derived from a bacterial adaptive immune system. It allows scientists to make precise modifications to DNA sequences. So here's a step-by-step breakdown of how it works. First, let's talk about designing the guide RNA, or gRNA. The core of CRISPR-Cas9's specificity lies in the guide RNA, which is a synthetic RNA molecule engineered by scientists. The gRNA consists of two main parts, a 20-nucleotide spacer sequence that is complementary to the specific DNA sequence you want to target in the genome. This sequence is what guides the Cas9 enzyme to the correct location. There's also a scaffold sequence, sometimes called Traxar RNA, that binds to the Cas9 protein. Careful design of the gRNA is really crucial to ensure it only targets the desired DNA sequence and avoids off-target cuts elsewhere in the genome. Next up, constructing the CRISPR-Cas9 complex. Once the gRNA is designed and synthesized, it's combined with the Cas9 protein. Cas9 is an enzyme that acts like molecular scissors, capable of cutting DNA. The gRNA and Cas9 protein assemble to form the CRISPR-Cas9 complex, which is specifically a ribonucleoprotein or RNP complex. The CRISPR-Cas9 complex needs to be delivered into the cells where gene editing is desired. There are, various methods that can be used, depending on the cell type and experimental goals. Electroporation involves using electrical pulses to create temporary pores in the cell membrane, which allows the complex to enter. Another approach is to use viral vectors, where the Cas9 and gRNA, or the DNA encoding them, are packaged into modified viruses, such as adeno-associated viruses or lentiviruses. that can infect cells and deliver the components. Lipid nanoparticles are also an option, encapsulating the complex within lipid vesicles that can fuse with cell membranes. Once inside the cell, the gRNA guides the Cas9 protein to the target DNA sequence in the cell's genome. For Cas9 to bind and cut, there has to be a specific short DNA sequence called the protospacer adjacent motif, or PAM, located immediately downstream of the target sequence. The PAM sequence, which is typically NGG for the commonly used Streptococcus pyogenes Cas9, is absolutely essential for Cas9 recognition and activity. When the gRNA finds its complementary sequence, and the Cas9 recognizes the adjacent PAM, Cas9 undergoes a conformational change and introduces a double-stranded break in the DNA. This break usually happens at a specific site, typically 3 base pairs upstream of the PAM. So when a cell experiences a double-stranded break in its DNA, its natural repair mechanisms jump into action to fix the damage. There are actually 2 primary pathways that the cell can use. The first one is called non-homologous end joining, or NHEJ. This pathway is, more common, but it's also often error-prone. Basically, the cell just ligates the broken DNA ends back together, which, sounds simple enough. But this process can lead to small insertions or deletions, what we call indels, right at the cleavage site. If these indels happen within a gene's coding region, they can cause a frameshift mutation, or even a premature stop codon. That means the gene can get knocked out or inactivated entirely. The other main pathway is called homology-directed repair, or HDR. This one is more precise, but it needs a homologous DNA template to work. If scientists provide an external DNA template, what we call a donor template, that has the desired new sequence and matches the regions on either side of the break, the cell can use this template to repair the DNA. This approach allows for precise insertions, deletions, or substitutions of specific DNA sequences, like correcting a disease-causing mutation or inserting a new gene. But just so you know, HDR is less efficient than NHEJ and is mostly active during certain phases of the cell cycle. After the gene editing process, it's crucial to screen and validate the cells to confirm that the desired genetic modifications have occurred. This typically involves DNA sequencing, which is used to confirm the presence of the intended edits and, to check for any unintended off-target edits. There's also phenotypic analysis, where we observe the cells or organism for any changes in their characteristics or function that are expected from the genetic modification. By manipulating these natural cellular processes, CRISPR-Cas9 has honestly revolutionized genetic engineering, enabling a wide range of applications in research,